Theft.
Oh. Good afternoon, everyone. This is our international lecture series 1 and topic 4. So continuously we are taking the lecture for the past 3 weeks, and last week we got a break, and this week we are starting again for the 4th topic. And I welcome you all on behalf of Nish Kanyakumari and Gambala University, Ethiopian. So today's topic is a novel approach to combat antibiotics resistance. So this will be conducted in association with the Department of Automobile Engineering, Noorul Islam Center for Higher Education, and Gambala University, Ethiopia. And our good speaker is Dr. Packia Lekshmi, Assistant Professor and Assistant Placement Officer, Department of Allied Health Science of Noorul Islam Center for Higher Education. And she is a renowned person for taking such a topic in biomedical stream. And I welcome Madam for this wonderful lecture. And without much ado, I like to invite her for this for her talk. Over to you, Madam.
Okay, thank you, sir. Am I audible, sir?
Yes, Madam, you are audible. So
Yeah.
if you have
Okay,
the
thank
slides, you can share it, Madam.
Okay, sir. Let me share. One second, sir. Yeah. Sir, yeah. Okay. Can you see my slide, sir? Sir, is it visible?
Yes, yes.
Yeah. Okay. Right. So greetings of the day. And before we dive into this topic, let me take a moment to express my gratitude.
Madam, you can put it in, Madam.
Yes, sir. Yes, sir. Yes, sir. I will.
You can put it in slideshow, Madam. From beginning.
Slideshow.
Others.
Yes, sir. Okay. So let me take a moment to express my gratitude to the management and organizers of Niche Kanyakumari and Gambala University, Ethiopia for giving me this opportunity. And special thanks to Dr. Lalu for inviting me to handle a session with this international winner. Thank you, sir. And today we are here to explore the topic, novel approach to combat antibiotic resistance. Do you know why I have chosen this topic? Because this antibiotic resistance represents a challenging global issue. That's the reason for selecting this topic, right? So not only the life science persons, our healthcare workers and the physicians should know about this particular antibiotic resistance, but every individual in this universe should know about this. It's our responsibility to lower the antibiotic resistance. Right? And considering this critical issue of increasing antibiotic resistance, there is an urgent requirement of identification and development, then validation and progression of novel strategies and approaches that can easily be utilized for overcoming this issue, global issue. So this is the code which is given by the World Health Organization regarding the antibiotic resistance. So it's a slow and silent global threat. It requires urgent attention, action, and international collaboration to prevent a post-antibiotic era. So see how much it causing a global threat. And do you know what is antibiotic? So we all are intaking antibiotics when we are getting infected with some kind of microbial organisms. So antibiotics are nothing but they are secondary metabolites, are the chemical, synthetic chemical, which is used to treat the infections, are used as a therapeutic agent. They work either by killing the microbial cells or by inhibiting the growth of the microbes. So these are the two ways the antibiotics act upon the microbial cells. So these antibiotics are designed in such a way to target the specific microbial cells and to destroy it. And resistant means, antibiotic resistant means, it is the ability of the microbe to kicking off the efficiency of its activity. Okay? So that is antibiotic resistance. So multi-drug resistant means the ability of the microorganisms acquiring resistant to more than one antibiotics. So some of the microbes are there which acquired, which developed resistant to more number of antibiotics. So if the microbe developed resistant to many antibiotics, how can we treat that? It's a problem. We have to go for some other treatment procedures or therapeutic procedures, right? Then coming to the reasons for antibiotics, how these organisms are developing, what are the reasons for developing this antibiotic resistance? One is over prescription and inappropriate use. Over prescription means nothing but excessive use of these antibiotics. For example, doctor may prescribe the antibiotic for some kind of illness for 5 days, but the person or the patient may take it for 10 days. So that is a over prescription. And inappropriate use means taking streptomycin for malarial infection, which is not a right candidate for malaria, right? And another example is treating the viral infection with bacterial drugs. That is inappropriate use. And second one is the incomplete treatment courses. How many of us are completing the whole course of medication which is prescribed by physician? Not all. Many of us are not completing the whole course of treatment, right? Skipping the intake of antibiotics when we feel better. I used to do that, right? Once I feel better, I used to skip the medication. I used to stop taking antibiotics. It's not right, actually. Right? You should complete the entire course of medication which is prescribed by the physician. And third one is the unnecessary use of antibiotics. So some people are using antibiotics unnecessarily. So for a headache or for some kind of small illness, if you take rest, it will be okay. But many people are taking antibiotics. So we should avoid this kind of unnecessary use of antibiotics. And the next one is the antibiotics which is used in agriculture. So in agriculture, for breeding and to get more yield in cattle farms as well as in poultry farms and in everywhere, they are using antibiotics. So these antibiotics, which will accumulate in those animals and birds and will pass on to the human beings as a resistant organisms. And the next reason is lack of new antibiotics. So we are in lack of new antibiotics. So that's why we are struggling in eliminating these antibiotic resistant microorganisms. So we have to go for searching these new antibiotics. We need to develop many new antibiotics to combat with this antibiotic resistance. And next one is global travel and trade. The best example is coronavirus. So this spread all over the world through travel and trade. So these antibiotic resistant microorganisms will also spread to entire world through travel and trade. So we should avoid transmitting these antibiotic resistant microbes to the entire world. And another reason is poor infection control practices. In many countries and in many villages, they are not following the hygienic practices. They are not following the infection control measures. So this leads to the development of antibiotic resistant organisms. And the last and important reason for antibiotic resistance is self-medication. So once in a while, we all becomes doctor, right? Yeah. We prescribe antibiotics for common problems, health illness, which occurs frequently. What we do means we just go to the pharmacist and we used to ask for so and so antibiotics. So without consulting the physician. So this is a main reason for developing this antibiotic resistance. Right? So these are some of the reasons. And coming to the mechanism, how these microbial cells are developing this character, antibiotic resistance, there are many pathways or mechanisms behind this antibiotic resistant development. One is the mutation. Mutation in the sense, when the microorganisms undergo genetic changes.
Madam, sorry to interrupt you. Slides are not moving. I think you have using the presenter view. So can you uncheck that presenter
One
view?
second, I will stop sharing and then I will share it.
Yeah.
1 second. One second. I share it again. Can you see it now, sir? Is it visible?
Yeah, now it's visible. Please
Okay,
uncheck
fine.
that presenter view.
Okay, okay.
Uncheck that use presenter view box and start from beginning.
Okay.
Or use the FY.
Yeah. Is it okay now?
No. Slideshow.
No?
Slideshow.
Slideshow.
Yeah, I'm in slideshow only.
From beginning, madam.
Yeah, okay.
Use that from beginning.
Right. Can you see it, sir? Is it visible?
No.
No?
No.
1 second. Yes.
Now it's visible, madam.
Is it okay, sir?
Yeah, you are using that presenter view. Uncheck that presenter view box.
Presenter view. No, I'm not using that.
The use presenter view is ticked. That uncheck that one. Under the monitor. Under the monitor. Next one.
Monitor.
Under the monitor.
No, I'm
above that monitors. Uncheck that use presenter view. Then it will come.
now. Now is it okay?
No, madam. You have to uncheck that use presenter view.
Check the presenter view.
After sharing, you can.
Yeah. So now it's shared, right? No.
No.
After sharing. And check.
Under the menu, madam, right end, there's use presenter view is there. No, below that.
This one?
Use presenter view. Uncheck that box.
There is no such presenter view.
Under the monitor is automatic. And
Yes.
that use presenter view is there. Uncheck that box. Is that visible, madam?
No, sir. I'm unable to find that presenter view. Sharing. No such view is there. Presenter view. The slide is not moving, right?
Yes,
That's
yes,
the problem.
yes.
Yes. 1 second.
Also, that slide one comes in full screen.
It's not coming in full screen.
Not coming in full screen. It is on side.
Oh, actually, I'm presenting. 1 second. Slide cursor is there. No, 1 second. Now is okay. I think now it's okay. I think now it's okay, right?
No.
Is it okay now?
No.
No. Actually, I unchecked that presenter view.
Okay.
Now,
Okay.
madam, you can use it from beginning. Yeah, that one.
Yeah. Now is it okay?
Yeah. Yeah. Okay. F5. Now F5.
Yeah, it's in presentation mode.
Okay.
Now it's in the first page, right, sir?
Yes. Yes.
Yes, ma'am.
Yeah. Do I want to repeat it from the beginning, or shall I continue it from that?
You can continue, madam.
Okay. Thank you. Coming to the mechanism of antibiotic resistance. So one is the mutation. So whenever this bacteria undergo genetic change that enable them to survive in the antibiotic containing environment, and this mutation will occur spontaneously, or it occurs naturally. And next thing is the plasmid. Plasmid is nothing but extra chromosomal element that carries many genes like a chromosomal genome. So some of the plasmid carries antibiotic resistant gene. If the bacteria containing the plasmid with antibiotic resistant gene, then that particular bacteria will resist against that antibiotics. Okay, for example, if the plasmid containing ampicillin resistant gene, that particular plasmid bearing bacteria will have resistance towards the antibiotic ampicillin. That's the thing. But there are many antibiotic resistant genes are there. Ampicillin resistant gene, streptomycin resistant gene, chloramphenicol resistant gene, rifampicin resistant gene. So many resistant genes are present in plasmid. If the bacteria bears these kind of plasmids, then it leads to antibiotic resistance. Then coming to the next one, horizontal gene transfer. You can see this one, this particular, this one. You can see the transfer of plasmid DNA, this plasmid which carries the antibiotic resistant gene. So this particular plasmid will be transferred to the
Madam, still
non-plasmid
the
area.
slide is not moving. Now we can see only the second slide. Can you move to your slides?
Okay.
Yeah, now.
Yes, now.
Now?
Yeah.
Now it's okay, right?
Yeah.
Yeah. So it's in the presentation mode. Sir, is it in the presentation mode? Am I audible, sir?
Yes, madam, you are audible. Now the
Is
slide
it
is on. Sound slide is
presentation mode. Okay, okay, okay. So see here, this particular, the first picture shows the horizontal gene transfer. Horizontal gene transfer. Here you can see the transfer of plasmid containing the plasmid which carries the antibiotic resistant gene is transferred to the bacteria which does not contain the plasmid. Okay, so it transfers. So these bacteria which carries the plasmid with antibiotic resistant gene shares a copy of those antibiotic resistant gene to other bacteria. So the colonies are the same strain of organisms will convert into antibiotic resistant strains, so that it spreads to entire population of the bacteria. Right? This is a horizontal gene transfer through conjugation method. There are few more methods are there to transfer the gene. Right? And this one is the blocking method. So in blocking method here, before adaptation, if we apply the antibiotics to the microorganisms, it will react on the microbes and it will kill it. But once it gets adapted, so like the usage of umbrella to protect us from rain and hot sun. So we are using umbrella, right? Likewise, these microorganisms adapt themselves to protect it from the antibiotics. It blocks the absorption. It blocks the intake of antibiotics inside the cell. So this is one mechanism. And the next mechanism is the efflux pump. In this efflux pump, in original, before acquiring the antibiotic resistance, this bacteria, whenever it intakes the antibiotics, whenever it eats the antibiotics, this antibiotics reacts with the bacterial cell and it kills it. So once it gets adapted, if the antibiotics enters into the bacterial cell and it's pumped out of the bacteria, it's called a flex pump. So from one side, that antibiotic is getting entered into the bacteria, and in another end, it's pumped out. Okay, so there will be no effect of these antibiotics. This is one kind of mechanism. And next mechanism is modifying the target site. So this is the normal one. Okay, so normal target site. So in this area, the antibiotics are prepared in such a way to bind exactly with the target site. Okay, so this antibiotic will bind in that target site and it will act on it and it kills the microbes. So see in the second picture, there is some modification or some kind of additional structure there in that target site. So these antibiotics cannot able to bind it. So if there is no binding of antibiotics in this target region, there is no action. So the bacteria survives there. So it's one kind of mechanism. And another mechanism is the breakdown. So this bacteria has the ability to produce many enzymes and proteins. Okay, these enzymes and proteins will destroy or degrade the antibiotics. Okay, so if there is no effect of antibiotics, the bacteria will survive happily in that environment. That's one mechanism of antibiotic resistance. And another mechanism is producing slime protection. So it produces an extra layer like what we do it in our real life. We used to construct an extra wall around our houses to prevent the intruders or the thieves. Likewise, these microorganisms produce an extra slime layer around it, which protect it from the antibiotics. So the antibiotics cannot able to enter inside, right? So it's another way of antibiotic resistant development. And how can we solve this? Okay. So there are a few solutions to solve this, to combat with the antibiotic resistance. One is by phage therapy. Phage is nothing but virus. Okay. Bacteriophage is nothing but virus which infects the bacteria and kills it, kills the bacteria. It's a natural process, right? So we are using that bacteriophage to kill the bacteria, the antibiotic resistant bacteria. We can use it as selective bacteriophages to kill the bacteria, and also we can go with these phage cocktails. Okay, mixture of phages. We can use mixture of phages, and we can use it as an elimination, use it to eliminate the bacteria. So that's one method. Another one method is these phage enzymes. So the phages are engineered in such a way to produce some kind of enzymes to destroy the bacterial cells. Okay? That is one way of treating this antibiotic resistant bacteria. And another way is an synergetic method. There's one synergetic way, that is combination of bacteriophage as well as antibiotics. So combining these two things in a synergetic way, we can kill the bacteria, antibiotic resistant bacteria. It's one method. And the next method is engineered phages. So normal phages are engineered to infect the specific bacteria or the antibiotic resistant bacteria. So specifically it is designed. Okay. So such bacteriophages, chimeric phages are used to destroy this bacteria. It's one way. And another one method is, it's also a combined procedure, bacteriophage along with CRISPR-Cas genes. So these two combine together and react on these antibiotic resistant bacteria, and it will eliminate it. It's one kind of solution. The next one is the CRISPR-Cas, a gene editing tool. So this gene editing tool is present in the bacteria, okay, or in some kind of actinomycetes too. So what it do means, this CRISPR-Cas, it recognizes the region where it requires modifications. In that area, it will modify the gene. So in the first mechanism, I said about mutation. So if there is any mutation occurs in the bacteria and the bacteria converted into antibiotic resistance, so that will be present in the gene. So the resistant gene will be replaced with sensitive one. That will be done by this CRISPR-Cas. So this CRISPR-Cas protein will be guided by guide RNA. Okay? There is a RNA which guides this CRISPR-Cas protein, and this
Starting from current slate.
அதுதான் கொடுத்தேன் நான்.
Current slate.
இதுதான் அதான் கொடுத்துருக்கேன்.
And the slide move.
Sir, is the slide is moving, sir? Dr. Lalu?
Yes, ma'am. Yes, moving.
Yeah. So this guide RNA will guide this CRISPR-Cas protein, and it will reach the destination point where it requires the modifications. In that particular area, it will modify it. Okay. So it will remove that resistant gene area, and it will be replaced with a sensitive one. Thus, we can convert this antibiotic resistant bacteria into antibiotic sensitive bacteria. So one method of solution. And next method is the combined therapy. So in combined therapy, so for example, So, for example, if if some person is coming forward to fight with us, that person is more stronger than us, right? Can we able to fight with him? No. So what we do means we'll go back, we will bring some of our friends along with us to fight with that stronger person. That's the reality. So similarly, here also, if one particular antibiotic is unable to fight with the bacteria, it is combined together with some other antibiotics, some 2 or more antibiotics, and these combinational antibiotics will fight against this bacteria and it will get destroyed. So one research was done by Brown, the scientist Brown. He tested on the trimethoprim resistant E. coli bacterial cells. Actually, this E. coli is resistant to the antibiotics trimethoprim. So what he did means, he combined 2 antibiotics, floxuridine and azidothymidine. And with the help of this synergetic drug pair, he reduced the bacterial load into 10,000-fold. Okay. See how the combined therapy is efficient. So in such a way, combining 2 or more antibiotics, we can eliminate the antibiotic resistant bacteria. The next method is immunotherapy. So in immunotherapy, by producing the antibodies is the natural product which is produced in our body itself. Whenever any antigens or any bacteria or virus or some other microorganisms, whenever it enters into our body, our defense mechanism get initiated. And this defense mechanism will produce some kind of molecules called antibodies. So these antibodies will fight against this bacteria or the viruses or the antigens, and it will eliminate it. So these are some kind of mechanisms which is done by the antibodies. So this is direct antimicrobial activity. You can see the Y-shaped molecule, which is antibodies, which will bind directly with the bacteria and it will neutralize it. And here, this is the virus. The spherical shape is the virus. These antibodies will bind with the viruses, the spike proteins, and it will neutralize it. And this is toxin neutralization. These antibodies will bind with the toxins and it will neutralize it. This is one kind. And another method is the opsonization. It's also this our cells, immune cells, along with the antibodies, binds with the bacteria or the viruses, and it will neutralize it. Another one method by immunotherapy is by activating the complement. So this complement, along with the antibodies, it will neutralize the pathogens which entered into our body. It's one method. And another method is by destroying the enzymes or proteins which promotes the antibiotic resistance. There are many enzymes or proteins which are responsible for promoting antibiotic resistance in the bacteria. For example, beta-lactamase. So beta-lactamase is an enzyme which is produced by the bacteria which has ampicillin resistant gene. Okay. So this beta-lactamase, what it do means, it will destroy the penicillin, the antibiotic, penicillin antibiotic. So if the antibiotic get destroyed, there will not be any efficiency. No, there is no more activity of these antibiotics. So by destroying these kind of enzymes, if we destroy this beta-lactamase enzyme, we can destroy the pathogen. Okay. So likewise, there are so many enzymes or proteins which promotes antibiotic resistance to the bacteria. And by destroying this, we can enhance the activity. And another one method of combating this antibiotic resistance is making new or novel antibiotics. We are lacking in this. Okay. There is a very big scope in drug discovery, finding new or novel drugs, a very big scope. Universally, we are having. So it requires interdisciplinary collaboration with a scientist, with a research persons, with a clinicians, with a pharmacologist, because they are going to produce the drugs, and other experts. So it's a collaborative work. With a collaboration, we can find more number of new or novel antibiotics to combat with the antibiotic resistant bacteria. So these are some of the mechanisms to combat with antibiotic resistant organisms. And this is one of the statement which is given by Dr. Arjun Srinivasan, who is the Associate Director of Healthcare Associated Infection Prevention Program at CDC, Center for Disease Control Center. So the era of antibiotics is coming to a close. In just 30 years, we will be back in the dark ages of medicine if we don't act. So it's better to act immediately. So I encourage each one of you to consider the role you can play in this critical issue. Whether you are a healthcare professional, or you are a researcher, or you may be a policy maker, or you may be an industry leader, or you can be a concerned citizen. Whoever you are, your actions matter, right? Let's continue to collaborate each other and innovate, or making new or novel drugs, and we can put our effort in finding new drugs. So in drug discoveries to combat with these antibiotic resistant organisms, where if we work in such a way, we can find a future where antibiotic resistance is no longer a threat, right? So together, not only we few people, the persons from the entire globe, we work together to fight this antibiotic resistance, so that we can prevent many illnesses which caused by these antibiotic resistant organisms. Thank you. Is there any discussions or doubts? Lalu sir?
Sir. Hello, sir.
Yes, madam.
Yeah.
Thank you, madam. Thank you for the wonderful presentation. And
Thank you, sir.
I think you have given a general instruction for all the antibiotic intakers. Because usually, if we get a cold, immediately we start taking without seeing the side effects of antibiotics, and we are falling into that trap, I think. By seeing this presentation, we are getting some awareness about the intake of antibiotics, and you are doing a wonderful research. And I think we can share that slides with our Gambala University team, and if they come across any of the questions, then we can answer through email. So
Yeah, sure.
if you possible, can share your contact details as well.
Sure, sir. Sure.
In the chat box.
Thank you.
Okay. So let me.
And I have shared that feedback link for submitting your feedback. So, as well as Dr. Packia Lekshmi has shared her details in the chat box. So, participants, please log on to that particular feedback form and give your feedback. And you will get a participation certificate for that. And you can get the details from Jagan Raj as well as our coordinator, Dr. Darwin Yeke. And thank you.
Thank you, sir. Thank you, Lalu, sir.
Thank you, sir.
And next
Thank
week we will continue with that topic 5.
Okay, sir. Okay.
Okay, thank you. Thank you.
Okay.
Shall we wind up?
Yes, sir. Yes, sir.
Okay, thank you. Thank you for everything.
Thank you, sir.